Okay, great. So talking about iron, there we go, lots of iron.
Again, this is my first MASTS meeting. So my name is Hepburn. I'm a postdoc working at the University of Stirling with Christian Schroeder. I've been working over the last couple of months also in collaboration with Ian Butler at the University of Edinburgh and Adrian Boyce at the Scottish University's Environmental Research Council, the NERC isotope facility based in East Kilbride. And together we have been working towards the development of a method that combines conventional sequential extraction procedures with Mössbauer spectroscopy to quantitatively characterize reactive iron mineral phases. The reason why we are interested in reactive iron in particular is because it's these phases that are bioavailable within the environment. These are the ones that are reactive enough to be used by biology. However, they are inherently difficult to analyze using standard conventional techniques such as X-ray diffraction because they are naturally present as amorphous phases or as very tiny colloids and nanoparticles. You've got two talks today from the University of Stirling, one by myself, and this project is specifically looking at the effects of the crystallinity, these amorphous problems on this issue. The other talk, which is again given by me, but on behalf of one of our PhD students, Deborah Wood, is trying to establish a technique which tries to reduce the effects of these small particles. So, because these particles are very difficult to analyse, in 2005, Simon Poulton and Donald Canfield came up with a sequential extraction technique that characterises different types of iron minerals into different types of chemically reactive groups. The purpose with any sequential extraction procedure is a series of chemical reactions that initially remove the most reactive phases. As you go on down the procedure, you target phases that are less chemically reducible using increasingly more aggressive techniques. The Poulton and Canfield protocol itself begins with a sodium acetate extraction which specifically targets the carbonate-bound iron. Easily reducible iron oxides, like ferrohydrate, are removed by a hydroxylamine hydrochloride bleach. Finally, the remainder of our iron oxides, acadionite, goethite, and hematite, for example, are removed by sodium dithionite. Now, the leach carries on to remove magnetite, silicates, and quantify the total iron available eventually. However, the first three stages are the ones that we are interested in. These are our reactive iron phases. Now, this technique has been used extensively in many high-impact studies throughout ocean and earth science, mainly to reconstruct early ocean chemistry and describe the major radiations in Earth's biological evolutions, and also to describe major processes in oceanic carbon cycling. Now, the purpose of this slide is not to go through each individual hypothesis. What I want to show you here is that each of the references in this slide is based on the Poulton and Canfield technique. Each of these references has over 100 citations, and the ones that are highlighted are published in Nature and Science journals as well. This work hasn't been just influential to lots of high-impact studies, it's also been applied to a large range of disciplines. Obviously, it's very important to the geosciences, but it very quickly has a high relevance to environmental, engineering, and technology applications as well. So, the Poulton and Canfield extraction procedure has been used extensively in high-impact interdisciplinary ocean and earth science over the last decade. And it has been used in this way and continues to be so because at the moment it is the only means that we have of physically isolating these different types of iron minerals. However, it is only semi-quantitative and it is not mineral specific. The most shocking or most worrying aspect of the technique is that it is significantly affected by varying degrees of mineral crystallinity, grain size, and organic matter complexation that is inherent of natural samples, this natural variation. In their original paper, Poulton and Canfield reported very high efficiencies for this method. However, even in their original paper, they noted that some of the stages were being prematurely removed within the sequence. So ferrohydrite is coming out in the initial stage rather than the second. Archegionite, a small amount, is coming out in the second rather than the third. And most notably, they saw that 5 to 7% of magnetite and 15% of nontronite were being removed before they were supposed to be. This is where Mössbauer comes in. The advantage of Mössbauer is that it is ion specific. The disadvantage of Mössbauer is that it is ion specific. So unlike ICP technologies where we can analyze a range of different elements, Mössbauer is specific to iron. But like I said, this gives it one main advantage because it has the ability, unlike the sequential extractions, to quantitatively characterize these individual ion mineral phases. This is a Mössbauer spectrum of a mixture of our less reducible iron oxides. So a mixture of archegionite, goethite, and hematite, which are all removed in that single sodium dithionite step. The Mössbauer can pick out each one, and the way that we quantify this is by calculating the area underneath each peak. Our experimental design was very, very simple. We synthesized a number of different iron minerals in the lab and supplemented this with natural materials, which have been very kindly donated by the National Museum of Scotland's Collection Centre. We mixed them all together, run them through the entire sequential extraction procedure, and at the end of each stage, subsampled and run each of these residual components through the Mössbauer to look at the difference between what minerals are present before and after each step. This is the Mössbauer spectrum of the complete mixture. In it, I've used one mineral from each step in the sequential extraction. Siderite represents our carbonate, our 2-line ferrohydrite represents our easily reducible iron oxides, and our goethite is representative of our slightly less reducible iron oxides. And it's these three that are our main targets for our reactive ion phases. So, running through the very first extraction, using sodium acetate to target the carbonate-bound iron, what we're expecting is the near complete removal of carbonate out of the siderite. What we see after the extraction is that the siderite is still sat in the sample. However, this isn't the most worrying thing. In comparison to the unleached spectrum, what we see is that the nontronite, this blue phase, has also been significantly removed in this initial step. Nontronite is an iron silicate. This shouldn't be targeted until the very last step in the procedure. The hydroxylamine step completely removes our ferrohydrite, so we didn't have anything unexpected in this stage. Moving on to our dithionite extraction, what we're expecting to remove is our goethite in this sample. However, again, we not only see that goethite is present in the spectrum, but that it's not significantly different from the original unleached phase. And not only this, the siderite that should have been removed in our first extraction step is still sat in the sample even after our dithionite. Now, what are the implications of this? Well, this is showing that not only are some of these stages ineffective, but some of the stages are not even targeting the exact mineral group that they're supposed to. This highlights significant issues with the accuracy and the reproducibility of these sequential extraction procedures, and it makes it necessary to revisit some of the most critical hypotheses that have been developed based on these techniques over the last decade in ocean and earth science. And it has major interdisciplinary implications as well. Thank you very much. Can I take any questions?
Great, thank you very much. Any questions from the floor?
Hello.
So, a lot of, by the way, great introduction. That really long sentence you gave was fantastic.
You stuck with it, yeah.
It was very technical. I really was going to say, repeat that, please. Great talk, and I can see that a lot of the literature has used this method, yet you presented a lot of worrying issues with this method. What implications does this have?
Well, for each individual hypothesis?
No.
Well,
just
in general, it means that the phase that you're expecting to remove isn't being removed. So if you're expecting that your whatever subsequent analyses you're doing, your isotopic measurements or any other microscopic or whatever work you're doing with that sample is based specifically on carbonate, it's not. It's also including all these other phases that aren't being removed. So the main implications are that up until this point, we haven't been looking at iron biogeochemical cycling, its impact on carbon cycling, and the use of these things as a paleoproxy in a mineralogical way. We've been looking at them in total iron concentrations and assuming that these extraction techniques and the concentrations produced from each one are specific to carbonate, specific to magnetite, specific to pyrite. If these are not correct, then, for example, use of this as a paleoproxy to define anoxic and oxic water columns means that these boundaries aren't lying where we thought they are. And in particular, in terms of the way that this links up with something like the evolution of life, this could be a good thing, this could be a bad thing. What we could be seeing is that these windows of big, large-scale oxidation events that are supposedly lining up with big radiations in biological evolution, either they're not going to line up at all, or one thing that it could show, it could help us to narrow this window down in a more positive scenario, and this would mean that we can focus these events even more. So either way, good or bad, hopefully it's going to help.
So there is room for improvement.
Yes,
Good.
yes, definitely.
Any other questions? Okay, great. Thank you very much, Laura.
Thank you.